Today I'll be discussing axon guidance, more specifically in visceral neurons. Okay. Before we dive into the aspects of the actual guidance of the axon, I want to highlight certain molecules and proteins that are essential for this. Okay, so starting off is DCC, okay? DCC. And with DCC, its target molecule, which I'll also put in green, and a nice little circle, is netrin. Okay, netrin. The other main pair, which I'll put in red, because of X, it opposes DCC, is robo. Okay, robo. There are 3 different kinds of robo. One, 2, and 3. That will be more prevalent later. Okay, but just like DCC, robo has a target molecule that I'll represent as a triangle called SLIT. All right. Lastly, the third protein, mainly I want to say is COM, which will be represented in a Y shape. Okay. All right. So now diving into the process of the axon guidance of commissural neurons, we have phase 1. Okay. Phase 1. This axon is new. It is forming and moving. Okay. Forming and moving. Alright. How it's moving is because DCC is attracted to netrin. Okay, so DCC is expressed across the membrane. So it's part of the intra and part of the extracellular. Okay, and it's being attracted to netrin. Okay, netrin is in a really high concentration by the midline. This is why overall commissural neurons soma might be on the left side, but they cross to the right because they're attracted to netrin, and so on. So DCC is attracted to netrin, high concentration by the midline. Okay, so actually I'll represent the midline as this orange of the line. What also is a high concentration is SLIT. Okay, SLIT is really high concentration by the midline. Okay. But during phase 1, the axon does not care about SLIT because because robo is inside the axon, held by com. Right? Com is holding on the robo so that only DCC has its active site extracellular for netrin to grab onto. Pulling this phase one, pulling this axon closer to the really high concentration. Okay, so that's phase one. Jumping into phase two, now this axon, actually erase some of these. Now this axon is coming in right to the midline. It's right in the thick of it. Okay. Still expresses DCC, right? DCC still getting expressed. But now, after several intracellular mechanisms that I won't be going into, robo is now outside. It's extracellular with DCC. Transmembrane. Right? And so, slit can now bind, as well as netrin, to their molecules. So I will take a snapshot of this, explain it a little better. Snapshot, come over here. Okay. We'll have membrane. Got DCC. Okay. With its netrin. And now, in this phase two, we have robo with slit. However, now that slit has bound to robo, this changes the conformation. Right, changes the conformation of DCC. We'll draw. Okay. Change the conformation of DCC, which inactivates DCC. Okay. Okay, so now we're switching from the attraction of DCC to netrin, inactivating the DCC. So now we're at propulsion. Propulsion with slit. So it's phase two. In midline, slit binds to robo, which inactivates DCC, and now we're at a repulsion stage. And the axon cannot turn around, right? It can't turn around. So it's going to be shoved. It's going to be shoved forward, repelled with slit and robo out of that high concentration, right? And if you can imagine it, this is the spinal column. It's crossing over the midline of the spinal column. So now in phase three is after the axon crosses the midline. All right. Comes over, crosses the midline. So this is when the different variations of robo come into play. If an axon expresses only robo 1, right, the robo 1, that variation, then it can turn closer to the midline, the utmost medial side, okay? Turns closer. 1. If the neuron expresses, if it expresses 1 or 3, okay, this can be confusing because it's not 1 and 2. So it's the way they were discovered. Okay, so it's 1 and 3. Then they'll turn a little bit after the 1. They'll turn that intermediate section. Okay. And if they express 1, 2, and 3. Okay, if they express, this neuron expresses robo 1, 2, and 3 in its axon, then it will take the most lateral route. It will turn the furthest away because there's a concentration gradient. So there's a high concentration of slit. This is a concentration gradient of slit. Okay. With more receptors that slit can bind to, it takes this out further to turn. Okay, so only 1, it'll turn closer to, only 2 of the variations it'll turn a little bit further, and with all 3 of the variations, it'll have to travel the furthest to turn. And these can turn up or down. Okay. Now, let me solve the bullshit.